The Mystery of Death and the Neurophysiology of the Last Hour of Life. The question of when exactly death occurs and what processes occur in the brain during these moments has perplexed humanity for centuries. Despite significant difficulties in obtaining reliable data on the state of the brain at the moment of death, recent scientific research has significantly expanded our understanding of these complex and enigmatic phenomena. Central to this quest have been the accounts of near-death experiences, whose experiences have provided a unique, albeit subjective, window into the dying process. They reported flashes of memory, out-of-body experiences, and journeys through a luminous tunnel, leading scientists to the idea of a special state of consciousness at the moment of death. These accounts, similar across diverse cultures and eras, provide valuable material for neurophysiological research. It is with them that we begin to build a bridge between human consciousness and biological processes. The renowned psychiatrist Raymond Moody described many such stories, which penetrated the public consciousness and sparked interest in the phenomenon of near-death experiences. Since his research, numerous testimonies have been collected, including from children, confirming the prevalence and recurrence of these experiences. However, a key question remains. Do these experiences truly indicate the preservation of consciousness after biological brain death? Or are they the result of specific processes in a living organ, albeit nearing its end? The answers to this question are still being sought, analyzing the functions of different brain regions and taking into account the effects of medications and metabolic processes. Clarifying the time frame of death became possible thanks to a rethinking of the criteria for its occurrence in medical practice. A key milestone was the Harvard Committee report, which, for the first time, introduced the concept of brain death, expanding on the traditional criteria for cardiac arrest. This radically changed the legal and medical approach to determining death and allowed the use of electroencephalography to record brain activity. Modern standards require the absence of brainstem responses, deep coma, and the absence of spontaneous respiration. However, even with clearly defined criteria, diagnosing death solely based on electroencephalogram data is difficult due to potential electrophysiological artifacts and ambiguous readings. At the cellular level, brain death is accompanied by a multitude of processes caused by a sharply reduced oxygen supply and the cessation of energy metabolism. Neurons lacking energy reserves quickly lose the ability to maintain ion gradients, leading to a massive influx of calcium and sodium ions into the cell. This causes cellular swelling, disruption of membrane function, and the release of neurotransmitters such as glutamate. The latter triggers a cascade of damaging enzymes, direct destruction of cellular structures, and the formation of free radicals, creating a vicious cycle of damage and leading to apoptosis. These events are accompanied by transient changes in gene expression, triggering a sequence of pathophysiological reactions that irreversibly alter the functional state of the brain. A significant source of information about the dying process has been the characteristics of neurophysiological changes observed during so-called near-death experiences. Subjects who have experienced these states describe flashes of memory, out-of-body experiences, a state of profound peace, and a unique clarity of consciousness that seems paradoxical given the critical decline in brain function. Scientific research aims to reproduce such states in laboratory settings, to identify neural correlates, and understand the mechanisms behind these phenomena. A key role here is played by brain oscillations of various frequencies, recorded using electroencephalography. The electroencephalogram EEG is an indispensable tool for studying brain activity, recording the rhythmic oscillations of neural ensembles. These oscillations are divided into ranges called delta, theta, alpha, beta, and gamma, each associated with specific functions and states of consciousness. Complex activity patterns provide clues to understanding the processes of perception, memory, and attention. Changes in the high-frequency gamma oscillation range, for example, are associated with conscious information processing, which is reflected in the study of the phases of brain death. An important detail is the recording area. Full-field 10 to 20 EEG systems provide a comprehensive overview of all brain regions, unlike the BIS device, which is limited to the frontal regions and focuses on monitoring the depth of anesthesia. Studies in healthy people have revealed that neural oscillations are not simply background phenomena, but perform active functions in supporting cognitive processes. For example, the alpha rhythm has long been associated with rest, but modern data highlights its role in suppressing redundant information. Theta waves are critical for memory and spatial navigation, while gamma rhythms are closely linked to awareness and attention. The interaction between alpha and gamma frequencies plays a crucial role in self-awareness phenomena, including the remarkable out-of-body experience, a feature of many near-death experiences. This effect is localized to areas of the parieto-temporal cortex of the brain, underscoring its importance in shaping the sense of self-integrity. A detailed study of brain activity during death revealed several sequential phases. In the early stages, after oxygen deprivation, a surge in beta and gamma oscillations is observed, reflecting the brain's attempts to maintain integrative function. This is followed by an isoelectric phase on the electroencephalogram, when neural activity appears completely suppressed, accompanied by the so-called death wave, pronounced membrane depolarization, and blockade of synaptic activity. However, surprisingly, upon restoration of oxygen supply, a wave of revival can occur, accompanying repolarization and the return of normal electrophysiological activity. These data challenge the definitive understanding of the moment of death and open up opportunities for prolonging life and rehabilitation after life-threatening conditions. Perhaps the most intriguing aspect is that despite the objective cessation of cardiac activity and the suppression of most brain activity, some neurophysiological processes can continue. This raises the question: is death an instantaneous event, or a process with a specific time course? Animal studies that allow for intracellular and cortical recordings reveal vivid dynamics in membrane potential and neural activity, providing valuable insights into this mystery. In clinical practice, it is beneficial to consider not only the heart, but also the brain for an accurate diagnosis of death, which is important for making ethical and legal decisions related to organ transplantation and medical interventions. The experiences of near-death survivors hold a special place because they combine neurophysiology with profound philosophy and subjective perception. Studies such as a large prospective study of patients recovering from cardiac arrest have found that many report elements of memory and awareness that defy traditional understandings of brain cessation. These data raise the question of the phenomenon of consciousness in the last moments of life and encourage the study of mechanisms that allow temporary maintenance of awareness in critical states. A significant challenge remains data variability, caused by differences in research methods, death models, and the brain regions where activity is recorded. For example, data obtained using a 2- or 3-channel monitor on the patient's forehead differs significantly from full hemisphere EEG recordings. Technical and biological artifacts, including muscle contractions and medication effects, can also impact data quality. This requires standardization of protocols, creation of comprehensive databases, and development of filtering algorithms to achieve comparable and reliable results. Finally, it's impossible to ignore the complex ethical implications and limitations imposed by modern concepts of life and death. Every step toward understanding these processes must be accompanied by deep reflection and respect for human life and its unpredictable boundaries. Studying the neurophysiology of dying not only helps unravel biological mechanisms, but also opens new horizons for understanding the nature of consciousness, memory, and, perhaps, human existence itself at the moment of its finitude. On this foundation, we can move forward to further explore the mysteries associated with the last hour of life and its reflection in the brain. Electrophysiology of consciousness and the phenomenon of near-death experiences. Human consciousness is a remarkable phenomenon, rooted in the depths of neural processes occurring in the brain. Neural oscillations, or rhythms, arising in neural networks, play a key role in the formation of perception, memory, and conscious experience. Their diversity is determined by frequency ranges, from slow delta and theta waves to fast-moving gamma rhythms. Each of these ranges is associated with specific cognitive functions. For example, alpha oscillations have traditionally been associated with a state of rest, but modern science points to their active role in suppressing irrelevant information, helping us focus on what's important. The discovery of beta and gamma rhythms has expanded our understanding of neurophysiology, demonstrating their connection to movement, attention, and conscious perception. Gamma oscillations, which emerged in the second half of the 20th century, are considered one of the most enigmatic rhythms. They correlate with awareness, the perception of the integrity of objects, and memory. It is precisely through the synchronicity of gamma waves that different areas of the brain can instantly coordinate information processing, creating a unified phenomenon of consciousness. However, questions remain. Can gamma oscillations serve solely as beacons of consciousness? Research shows that such rhythms occur both during sleep and under anesthesia, when consciousness is clearly diminished, adding to the complexity of their interpretation. The key example is the paradox of their intensification during deep, rapid eye movement sleep, when the brain experiences an information-rich internal world. The study of memory is also closely linked to neural oscillations. Theta rhythms, first observed in the hippocampus, are closely linked to the formation and retrieval of memories. Remarkably, the same rhythms are activated during meditation as during memory processing. The interaction of theta and gamma waves is believed to be the basis for encoding and storing episodic events in the brain, making them fundamental elements of our self-identity. This neurophysiological dance of rhythms allows us not only to remember, but also to feel, to create coherent stories of our lives. The phenomenon of near-death experiences, NDEs, reflects a combination of many of these patterns. People who have experienced a critical near-death experience describe vivid memories, sensations of separation from the body, and even journeys into light-filled tunnels. These experiences are associated with bursts of activity in the alpha, theta, and gamma ranges, demonstrating a complex neurophysiological picture of consciousness under extreme conditions. Such survivors often report a reduction in the fear of death and a reappraisal of values, a shift from a materialistic to a spiritual perspective. This raises the question, perhaps the brain is using its last resources, creating a powerful phenomenon of consciousness before the end. Animal and human studies confirm the presence of brief bursts of high-frequency activity in the brain during dying. For example, in rodent experiments, enhanced gamma waves were recorded after cardiac arrest, persisting for several tens of seconds. One hypothesis suggests that this is the neurophysiological trigger for the last act of consciousness. Despite the cessation of blood circulation, the brain is capable of briefly maintaining activity, possibly providing a subjective experience associated with memories or a state of rest. This seems paradoxical, since death is traditionally associated with the absence of any brain activity. Researchers also faced a fascinating question. When do we actually die? When the heart stops, or when brain function ceases completely? Modern medicine relies on complex criteria, including the loss of consciousness, brain reflexes, and breathing. EEG helps record the brain's electrical activity, but it may not disappear immediately, but over a long period, sometimes up to several hours. Of particular interest is the so-called death wave, a period when a sharp disruption in electrical activity occurs, which can be followed by a rebirth wave when oxygen returns. These cycles indicate that brain death is a complex, multi-stage process. Research into the neurophysiology of NDEs reveals the importance of a brain structure responsible for spatial self-awareness, the parieto-temporal junction. Disturbances in this area lead to conscious out-of-body experiences during critical states. To the layman, these phenomena appear paranormal. But from a scientific perspective, they are a manifestation of a disruption in the integration of sensory signals caused by specific brain rhythms. Alpha rhythms are enhanced, isolating the body from external perception, while gamma oscillations activate internal images and out-of-body experiences. Despite significant advances, much remains unknown in the study of brain activity in the dying. Differences in measurement methods, physiological characteristics of different organisms, and complex ethical issues pose challenges. Studies based on subjective descriptions of NDEs remain an important source of data for linking behavioral and neurophysiological manifestations. With new technologies, there's hope for a more complete understanding of how neural activity generates conscious experiences at the threshold of life and death. Understanding the mechanisms of consciousness and memory formation during dying not only advances fundamental science, but also opens up potential for clinical applications. For example, improving resuscitation and neuroprotection capabilities in critical conditions could be based on understanding the precise phases of neural changes. Moreover, this information sheds light on philosophical and existential questions of human life, bordering on the fine line between existence and non-existence. Moving on to the next aspects of the study. It is important to focus on episodes of functional recovery of neural activity after critical damage and the possibilities for its correction. This will allow for a better understanding not only of the phenomenon of death, but also the conditions under which consciousness can be restored or preserved despite severe brain damage. Problems of brain research, near death, and ways of standardization. The study of the neurophysiology of death faces numerous challenges, leading to significant discrepancies in the data from various studies. First, the method of recording brain activity plays a crucial role. For example, full-scale 20-turn electroencephalography, EEG, and limited recordings, such as those made with a binaural index monitor, are used. The former covers a wide range of brain regions, while the latter records electrical activity only in the frontal lobe, leading to significant variability in results when compared. Furthermore, available methods have varying sensitivities to neural activity at different frequencies, further complicating the interpretation of the data. A significant portion of the discrepancies in the studies is explained by the modeling approach used. Experiments are conducted on different animals using different methods of inducing death, from decortication and potassium injections to hypoxia and cardiac arrest. Each method elicits unique physiological and biochemical responses, significantly influencing patterns of brain activity. Furthermore, the duration of observations and the depth of recording also vary, altering the chronotype of the recorded activity. Animal experiments, unlike clinical observations of patients with various neurological pathologies, allow for more controlled conditions, but their results are not always transferable to humans. Human research faces numerous additional obstacles. Scientific study of the brain in dying patients is complicated by ethical and technical considerations. Patients included in studies always suffer from some form of illness, trauma, stroke, or chronic disorders. These comorbidities alter the neurophysiological picture, taking into account various treatment strategies and medications, as well as anatomical and functional damage. It is not uncommon for drugs with sedative, analgesic, or anticonvulsant effects to be used, which can distort the electrical activity of the brain and increase the discrepancies in interstudy data. Particular attention should be paid to the factor of drug influence. During seizure control or resuscitation, a variety of drugs are used that alter metabolism, vascular tone, and synaptic transmission. Anti-epileptic, sedative, and narcotic substances affect the rhythmic activity of neural networks, often suppressing gamma and alpha oscillations or inducing pathological patterns. The goal of researchers is to identify and minimize the impact of these contaminants. However, excluding such patients from samples is often impossible due to the limited available data. Technical aspects such as motion artifacts, electrical noise, and equipment malfunctions must also be considered. High-frequency noise in the gamma range is sometimes caused not by neuronal activity, but by muscle contractions or interference from the electrodes. These distortions can easily be confused with genuine brain patterns. Therefore, the development of signal cleaning algorithms and standardization of recording procedures are of paramount importance for improving data quality and reducing variability in results. Furthermore, methodological variability also affects the time and location of measurements. The timing of recordings of dying brain activity can coexist with the phases of arterial pulse, respiratory rhythms, and responses to external stimuli, and neural networks in different brain regions deteriorate at different rates and in different sequences. Topographic features also play an important role. Some researchers record the electrical energy of the cortex, while others record the activity of deep structures, such as the brainstem or hippocampus, which leads to incorrect interpretations of the dynamics of brain death. The use of animal models is ideal for standardization efforts, but the scalability of data to humans has its limitations. For example, results obtained in rats or macaques are difficult to extrapolate to the complex human brain with its intricate interregional connections and cognitive functions. Therefore, combining clinical observational data with controlled model experiments requires rigorous selection and careful comparative analysis. Standardization of studies is a necessary step to overcome these difficulties. Developing a unified recording protocol that captures electrode placement parameters, observation timeframes, and experimental conditions will help significantly reduce discrepancies. Specifically, the introduction of a unified neurophysiological minimum set of measurements is recommended, including multi-electrode recordings with simultaneous monitoring of medication status and physiological parameters. Coordination of interdisciplinary teams also plays a key role. Collaboration between specialists in neurology, neurophysiology, critical care medicine, and biomedical engineering can provide a multifaceted view of processes in the dying brain, while systematization of data collection and analysis methods will help establish representative biomarkers of neurophysiological death. Furthermore, the creation and exchange of publicly accessible databases containing complete EEG recordings from patients, performed according to established standards, will unite the efforts of international researchers and advance neuroscience in the study of death to a new level. This will address the lack of information due to the rarity of cases, as well as the diversity and specificity of the pathology. Difficulties can also arise when collecting subjective descriptions of experiences related to dying. Although accounts from near-death survivors provide valuable information about consciousness during the dying process, integrating them with objective physiological data requires careful selection and interpretation. The development of standardized rating scales, such as the Near-Death Experience Rating Scale, NDES, and their alignment with physiological markers facilitates more accurate data comparison. Taken together, the current state of affairs demonstrates the complexity and multidimensionality of studying the neurophysiology of death, but also points to solutions. Unified protocols and standards will reduce data variability, improve the reproducibility of results, and, consequently, expand our understanding of what happens in the brain during the final moments of life. The development of structured studies that control all key parameters, including study design, recording type, location, and medication status, will become the foundation for developing a comprehensive picture of brain death. Ultimately, this will ensure not only scientific advancement, but also a significant contribution to the ethical and clinical understanding of death. Moving on from an analysis of the factors that cause discrepancies in the studies, the next step will be an in-depth examination of the neurophysiological processes detected directly at the moment of the transition from life to death.